Namaste. This is the Professor, CBMD PhD, for today's Saturday, October 21st, 2023. It is 10:33 and 43 seconds, a.m. Eastern Standard Time, and today's research, Glioblastoma. So let's jump to it here. Researchers assign gene therapy that can effectively target glioblastoma. Date October 18, 2023. Source: Brigham at Women's Hospital. Glioblastoma, an aggressive brain cancer, is notoriously resistant to treatment, with recurrent glioblastoma associated with survival of less than 10 months. Immunotherapies which mobilize the body's immune defenses against the cancer have not been effective for glioblastoma, in part because the tumor surrounding environment is largely impenetrable to assaults from the body's immune system. And to convert this immunosuppressive environment into one amenable to an immune response, investigators engineered an oncolytic virus that can infect cancer cells and stimulate an anti-tumor immune response. The results demonstrated the safety and preliminary efficacy of the gene therapy approach in high-grade glioma patients with prolonged survival in a subgroup of recurrent glioblastoma patients immunologically familiar with the virus. Glioblastoma has an aggressive effect, in part because of a multitude of immunosuppressive factors surrounding the tumor, which enable the tumor's growth by preventing the immune system from entering and attacking it. And the study showed that with a virus they designed, they can reshape the immune desert into a pro-inflammatory environment. The Phase 1 first-in-human trial examined the safety of an oncolytic virus called CAN-3110, which was designed and subjected to preclinical testing. The researchers at BWH had licensed to Candel Therapeutics as the trial was ongoing. The cancer-attacking virus is an oncolytic herpes simplex virus, which is the same type of virus used in the therapy approved for the treatment of metastatic melanoma. Unlike other clinical oncolytic herpes simplex viruses, this therapy includes the ICP34.5 gene, which is often excluded from clinical oncolytic herpes simplex viruses because it causes human disease in unmodified forms of the virus. However, the researchers hypothesize that this gene may be necessary to trigger a robust pro-inflammatory response necessary for attacking the tumor. Therefore, they designed a version of the oncolytic herpes simplex virus, one that contains the ICP34.5 gene, but is also genetically programmed not to attack healthy brain cells. Overall, the trial demonstrated the safety of CAN-3110 in 41 patients with high-grade gliomas, including 32 with recurrent glioblastoma. The most serious adverse events were seizures in 2 participants. Notably, glioblastoma participants who had pre-existing antibodies to herpes simplex virus 1 virus. 66% of the patients had a median overall survival of 14.2 months. In patients with pre-existing antibodies, the researchers saw markers for several changes in the tumor microenvironment associated with immune activation. They hypothesized that the presence of herpes simplex virus 1 antibodies resulted in a rapid immune response to the virus, which brought more immune cells to the tumor and increased the levels of inflammation in the tumor microenvironment. After CAN-3110 treatment, the investigators also observed an increase in the diversity of the T-cell repertoire, suggesting that the virus induces a broad immune response, perhaps by eliminating tumor cells, resulting in the release of cancer antigens. These immunological changes after treatment were also shown to be associated with improved survival. But studies like this one show the promise of gene therapy for treating intractable conditions, the scientific discoveries made by researchers in the first-in-human clinical trials, and, ultimately, life-changing treatments for patients. The Institute's multidisciplinary approach sets it apart from others in the space, helping researchers to rapidly advance new therapies and push the technological and clinical boundaries of this new frontier. Going forward, the researchers plan to complete prospective studies to further investigate the effectiveness of the oncolytic virus in patients who do not have antibodies to herpes simplistic, virus 1. Having demonstrated the safety of one viral injection, they are proceeding to test the safety and efficacy of up to 6 injections over 4 months, which, like multiple rounds of vaccination, may increase the effectiveness of the therapy. The new 6 injection trial is funded by Breakthrough Cancer. Almost no immunotherapies for glioblastoma have been able to increase immunoinfiltration to these tumors, but the virus studied here provoked a very reactive immune response with infiltration of tumor-killing T cells. That is hard to do with glioblastoma, so their findings are exciting and give us hope for the next steps. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.